we're starting with the next part, which is the once we have seen in the previous chapter, we have seen all the molecular mechanisms of how the genome is replicated, how the mRNAs are made. So the mRNAs are made into the proteins, and now you have the genome RNA made and the proteins for the virions which are going to come out. That is also the components for those are also made. So for any successful viral infection, all right, so any successful viral infection that will require that after the initial cell is infected, it needs to go to the next one or basically disseminate itself into the host. And for that, you absolutely require them to make progeny virions. We all know that what does that mean? It will just produce a lot of them. And now we have all the components for that is synthesized. And here we are going to see the first one is that how all these different components are trafficked and assembled together. Now, what we are going to see that there will be several different types of things that's going on, and where the confusion occurs, so that remember that they have to package the viral genome, which could be DNA or RNA, that will interact with proteins. Now, many times what happens, you see a slide, you will see that you have a bar right here with some of the motifs. Here you see a bar with the motifs. And sometimes it's like you get confused that what is what. Just make sure you carefully look at that where you have a DNA or RNA, and which is both look like bars and some of the sequences, but just read the material and see that, okay, all right, this is the protein, this is the DNA or RNA. They are going to interact with the specific domains and going to get packaged into the virus. So this is one of the key points that make sure that when we come to those, you just check on those. So again, the structure of the virus particle is basically determines that how it is formed. And what we mean by that, there are lots of naked viruses, and you can see those naked viruses will not have an envelope, whereas the enveloped virions usually will bud out at a certain part, not necessarily plasma membrane. It could be nuclear membrane, endoplasmic reticulum, Golgi intermediate complex, or even Golgi. So any of the endomembranous part, they will acquire that envelope and then they will bud out. And depending upon the complexity, we are going to see that the viral structure basically detects or tells that how it is basically formed. So here is a general scheme, which is the complete set of common reactions that basically you have. Very self-explanatory, formation of structural units of the protein shell for one or several protein. One protein will be just the simplest one, like a parvovirus, where you have just one capsid C protein. That's it. Other than that, every other virus will have multiple protein. And in some of the cases, several of them, many of them, if not hundreds. Probably hundreds plus in the giant viruses. And the next part, what we are looking at, is the assembly of the protein shell by appropriate, sometimes variable interactions among the structural unit. We're going to see, and if it is an icosahedral symmetry, and if the T number is more than 1, we know that there will be these quasi-equivalence positions, because they need to form these pentons and the hexons. We are going to use that part all the time, that viral assemblies, which much more complexity of the icosahedron are going to form by pentamers and hexamers. So the pentamers will be called as pentons. Hexamers are going to be called as the hexons, which are going to be made with different combination of proteins. And then you have the acquisition of an envelope for the acquisition of the envelope for the enveloped virus. This is common to many viruses. And release of the host cell is also universal. On the other hand, the maturation of the virus particle is also common in many viruses, but not all. So only the blue things are a little bit selective. What we mean by the maturation of the viral particle, say for an example, if it's a naked virus, many of the naked viruses, as soon as it is released from the cell, it is fully infectious. On the other hand, in some of the enveloped viruses, what happens that still some of the maturation, still some of the proteins are in its pre-form. That means that the proteins have not fully matured. It takes just several seconds to a couple of minutes for that virus to be matured, and that's what we mean by that, and we'll explain that process. towards the end of the class. So here, the picture is a very general picture of you have seen that there's polyomavirus, and it's a general idea. But what we are looking at, the assembly is dependent on the host cell machinery. And what are the main players? First is the cellular chaperone proteins. These assist the proteins of the host to fold properly. Once the protein comes out of the ribosome, it is in a nascent form. It must have to form, or they will automatically form these alpha helices and then beta sheets. But that's not all. Even after the alpha helices and the beta sheets, which is a secondary structure, they must fold into a three-dimensional tertiary structure. And many of these protein folding is assisted by the cellular chaperones. And one of the small things is that why these chaperones are needed, there are some hydrophobic residues on these proteins which are coming out of the ribosome. Now, these hydrophobic residues are extremely unstable in the aqueous environment. Chaperone proteins immediately bind to those residues and fold them in such a way, that's the native fold of the protein, that these hydrophobic residues actually are buried inside the protein, and the protein actually becomes, after that, very stable in the aqueous environment. Hydrophobic residues in aqueous solution or environment cannot be exposed towards the outside. The structure will fall off. Then you have the transport system. Now, transport system, many different types we're going to see in a second in the next slide. And then you have the secretory pathway. Now, what I did that I teach this in the cell biology class, and I have put my two supplementary videos in the top section of this module in there. And you really do not need to go into the detail, but just have an overview of that the proteins which are basically destined, particularly to the plasma membrane or secreted from the outside or going to the lysosome, they are basically synthesized on inside, either if they're soluble inside the endoplasmic reticulum, or if they are membrane bound, they are formed on the surface of the ER membrane with the associated ribosome. I'm going to go into a little bit more detail of that. And this pathway where you have the polyribosomes are coming onto the ER, synthesizing the protein, either membrane or soluble protein. Then they go into the endoplasmic reticulum for further modification, loss of glycosylation and protease cleavage and those things. And from the Golgi, they are transported, usually for the virus, most of the time towards the plasma membrane. But remember that some of the viruses can assemble from the Golgi or the ER intermediates as well. So that's the secretory pathway. So what is not the secretory pathway? That's the question. So secretory pathway, you always think that protein is going to be secreted. It's not that. Plasma membrane proteins are all synthesized in the secretory pathway. So it's not that the proteins which are synthesized in there are just only secretory proteins. So the non-secretory pathway is that if it is a cytosolic protein for the host, which is produced in the cytosol, they are produced by the free ribosomes in the cytoplasm. And other pathways do exist. Protein can be transported to the mitochondria, protein can be transported to the nucleus, and all of those basically are non-secretory pathway. But just remember that for the cell, lysosome, cell membrane, and outside secretion, these are the pathways which is utilized by the secretory pathway. And then we are going to see the nuclear import and the export machinery as well. And you can easily see right here in the picture that LT and the ST, small and the large T antigen, and the large T antigen is exported inside the nucleus, because remember that all of these proteins, whatever the virus needs, they're all synthesized in the cytoplasm. And many of them will be needed inside the nucleus. So you need a nuclear import and the export machinery. Where you need so these are all imports. Where you need the export, you need the genome or the associated genome and the proteins to be exported outside, because it needs to be released from the cell. So you need all of these. So here is moving in the heavy traffic. And what happens in this one, or what we trying to explain, that the cytoplasm is almost like a gel-like consistency, and nothing happens in there very fast. It's not that the diffusion can happen extremely fast for the proper assembly or movement of the viruses. So how do you move in the heavy traffic? So for short distances, which is angstrom to nanometer distance, usually what you have that these are present on the plasma membrane, nuclear membrane, ER, and the Golgi membrane. These are usually by pores, portals, translocons, these type of structures. So the translocon, this typical structure, what is in there is a translocon. So translocons are the channels which are present on the endoplasmic reticulum, through which the nascent proteins which are produced by the ribosome can get inside the lumen of the ER. So those are called the translocons, small. If it is a secretory protein, the whole protein will go into the ER lumen and remain in the soluble form. But what if it is a membrane-bound protein, the translocon will pass a part of the protein, and then from the translocon is going to shift that protein onto the membrane, recognizing some hydrophobic sequences. So you will have a tail or a head part of the protein. Head or tail means that the N-terminal or the C-terminal hanging into the lumen. You have an intramembrane or transmembrane part. Then you have a head or tail sticking it into the cytoplasm. So these are done by the translocons on the ER membrane. And you are going to see that nuclear pore and the portals, very elaborate structure there, and there are lots of channels that we have seen in the membrane structure as well, plasma membrane. Then the long distance are the energy-dependent motor proteins on cytoskeletal tracts, and there are two types of filaments that are used for these. There are superhighways, and then there are smaller roads. The superhighways are made up of microtubules. And on those, there are two types of motor proteins, dynein and the kinesin. They actually move on the superhighways or the microtubules. And again, these are polarized, have a plus end and a minus end. The same way, the microfilaments are made up of actin. Again, I will repeat this one if you have not taken cell biology. So the microfilaments are made up of actin. Microtubules are made up of tubulin. And they polymerize, depolymerize, and these are the cytoskeletal structure. Now, you have two different types of motor proteins that utilize these highways or these structures to move onto them. If it is a microtubule, the motor proteins are the kinesins and the dyneins. They walk on these. If it is a microfilament, actin-based, the motor proteins are the myosins. So this is an overview of that, and you will see that the viral structures move by this. So this is a next classic experiment, and this is, we can ask some critical question by modifying this experiment basically a little bit. So what you are looking at, that here a cell, which is the DAPI stained nucleus, blue you can see, and the red is an anti-tubulin antibody. Tubulin are the monomers, there are alpha and beta tubulins. Those are the monomers which make the microtubules. So the red part is the cytoskeletal, just only microtubules, the anti-tubulin antibody. And now this experiment, what was done, they took the VSV, vesicular stomatitis virus, which contains the P protein or the phosphoprotein. So this guy, Shubhasi Das, I'm going to talk about him a little bit later, why I have a picture of him. So what he did, that he took the phosphoprotein and fused and enhanced GFP to that. That's it. So you have a fusion protein, VSV, P protein with a fusion GFP. Now you have a recombinant virus, which is functional. Even with the GFP fusion, the virus can actually work. And so now what you see that in the real time, when you are looking at the virus, what you are looking at that all these yellow and the green spots are towards the plasma membrane. It's not really a viral factory because you see it towards the top, towards the side. One side is just more of them. They are budding out of the cell. These VSVs, vesicular stomatitis virus, budding out of the cell. And what they do, that they utilize these microtubule highways to get to the plasma membrane. How do you know that? The bottom experiment is the same experiment, But what they did, that they added a drug, nocodazole, and it's all in your notes. They added the drug nocodazole into the Petri plates, and the nocodazole will basically dismantle or disintegrate the microtubules. Now what you see the aggregates of the virion all throughout the cytoplasm. They fail to reach the plasma membrane for budding. So this is a guy, he was in Nebraska Medical Center at the time when it is. I know this guy from undergraduate. We went to undergraduate. We went to college together. So he is now in the senior scientist in Takeda Pharmaceuticals in the vaccine development. They work on Zika, dengue, these type of viruses, flaviviruses. So that's what it is. It's a personal friend of mine. Very, very seminal paper. So he presented this one in 2005 at the Penn State, where we had the American Society of Virology meeting. This was the paper that he presented there and published next year. Alrighty. So this is what we told, that nothing happens fast in the dilute solution. You can see that the viral components often visible by light microscopy. These are called the factory or the inclusions. We have already seen that in the some of the beginning chapters. And they concentrate proteins on the internal membrane that we have already seen. Like the poliovirus, they make these RNAs on these double vesicles. Then we have seen that, particularly when we did the special, the special topic on coronaviruses, that they make up these double membrane vesicle in which they have the RTC or the replication transcription complex are made up and set up inside those. And here what we are looking at, that on the right-hand side, you can see that there are some dark bodies in the cells, and these dark bodies are called the Negri bodies, which are the viral factories, and this is a signature rabies virus. And if somebody is infected with that, this is what they will show. And these are some of the structures that you can even see without an electron microscopy, basically. So basically, what is the take-home message is that there are certain spaces in the cytoplasm where the assembly actually occurs. It's not so for that, basically what you need to bring the components to assemble to certain areas of the cytoplasm or the nucleus or certain areas of the cell. That's what we mean by that nothing happens in the fast in a dilute solution. They are not scattered all over. And how they get these things together, we are going to see that. So how these viral proteins actually know where to go, because you have to make a structure, you need the proteins in there to build that, how they know this. So they know this by some specific sequences. And for the membrane targeting, one of these are called the signal sequences. Now, again, remember that if you're just using the secretory pathway, at the N-terminal, the N-terminus of the proteins, if they're soluble, has a signal sequence, which is cleaved off. That's number one. Number two, that if you have a membrane protein where the N-terminal is basically facing the membrane, let me just quickly draw this because so suppose this is your ER membrane, and here you have a ribosome sitting onto the translocon. If there is a protein that is soluble, goes into the ER lumen, your signal sequence is right here, and this is chopped off. So this is your N-terminal. So this is your N-terminal, this is your C-terminal. This will become the N-terminal in the matured protein. Now, if it is an N-terminal protein right here, where you have the, right here, N, this is C. If the N-terminal of the protein is in the lumen, you also have a signal sequence. But in addition to that, you must have a signal for the membrane retention, which is somewhere here. So that's one of the things. And this will bring that protein in here. So the matured protein will look like this. So you're looking at this protein. The matured protein will look like this. This is your N-terminal, and this is your signal, membrane retention signal, and this N-terminal is chopped off. Why I'm just putting this? Because if you have a protein which has the N terminal on this side and the C terminal on this side, those proteins will not have a signal sequence in there. And these are, so there are different types. I just put that if you have a type 1 membrane protein, it will have an N-terminal signal sequence. Other than that, you will have these membrane sequences all over the place. So, so that is your membrane retention signal. And then there are glycosylation fatty acid modifications. These are also used for the addresses. Along with that, there are some specific signal like nuclear localization sequences or NLS. This, if it is present on the protein, they are going to be exported into the nucleus. Remember that. They are going to be exported into the nucleus by the nuclear pore. That's what it is. We have seen that. On the other hand, they are going to be imported into the nucleus if they have a nuclear localization symbol. They are bringing it in, import. Nuclear export signal means that once, say for example, protein components are associated with the genome, like a DNA or RNA, if they are needed to be exported, then you need an export signal as well. So you need an import signal. Usually the import signals are recognized by some of the adapter proteins, which we call importin alpha or importin beta. They interact with the nuclear pores to bring things in. That is your nuclear localization signal, NLS, recognized by importance. Whereas the nuclear export signal, these are recognized by a class which are called the exportins. Again, specific names are in there. Exportins which export basically these proteins or the protein and the genome outside of the nucleus. So here we are looking at two examples of the NLS, the third in the row. So here you are seeing that you have a proline followed by a stretch of basic amino acids, 3 to 7, and then followed by another protein like a valine in there. This is the SV40 T antigen. This is a nuclear localization signal. The T antigen must go to the nucleus for doing its function. On the other hand, nucleoplasmin is also another protein which has a bipartite NLS. So what you are looking at, that you are looking at a stretch of basic amino acid, then random amino acid, and followed by another stretch of basic amino acid. So see that the lysine arginines are in there, and you have these random amino acids in here. So that's basically what the sequence looks like in the nucleoplasmin. Again, you do not have to memorize that. Just to remember that you have a proline followed by some basic sequences, hydrophobic sequences, and then followed by hydrophobic sequences like this. And you have a basic followed by random sequences, followed by another basic sequences. These all work like that. And there are many other modifications of these. So here we are looking at that how you put the viral proteins within the nucleus and where they are coming from. So let's start one at a time. So here is the viral protein, which is the influenza virus nucleoprotein, the nucleocapsid protein. This will enwrap the genome RNA. So this will be the nucleocapsid protein. So they are synthesized in the polyribosome, in the cytoplasm. They are not membrane proteins. And then they are imported inside the nucleus where they will basically enwrap the RNA. On the other hand, here we are seeing that this is a polyomavirus VP1 pentamer. All the colored structures are the 5 subunits of the individual subunits of the VP1, so you have all, not the subunits, or the monomers of the VP1, 5 of them. And in the middle, just like the dot, a greenish, grayish color or blackish color dot in the middle, this could be VP2 or VP3. And again, the pentamers, there are 2 different types of pentamers in polyomavirus. And if you just remember that, they actually do not follow the triangulation number properly. So it's an odd example of that, but they have 2 different types. But anyway, they are imported also into the nucleus as these pentamers. You need to have formed these preformed pentamers with the VP2 or VP3 before they can be imported. On the other hand, this is an example of adenovirus. What happens that the first, you have these hexons are basically a trimers of these hexons are going to assemble together, and the L4 protein of the adenovirus help in this maturation of these hexons. And then also the protein, which is called protein pre-6, basically associate with some of the important molecules to import these hexons into the nucleus. We're going to talk about these in a little bit more detail, that how these occurrences are basically happening. So this is basically the localization of viral protein within the nucleus. And all of them, remember that, will have to have these nuclear localization signal, which is the third part of this. And then we're going to talk about the others as well. This one, location of the viral protein to the plasma membrane. So the first one, remember that, membrane targeting. Signal sequences, glycosylation, fatty acid modifications, and membrane retention signal. So the first 1 and 2 we are going to talk about in this one. So here we are looking at the secretory pathway and how this happens. You can see that when a viral protein or any protein in the secretory pathway, when it started to form by the ribosomes, those ribosomes are brought into onto the membrane of the ER. So here you are looking at a nascent polypeptide chain or a protein, of course a viral protein, that is being translocated into the membrane. And this is a membrane protein. That's why this right here, the portion that is spanning the membrane, will have that blue stretch. That is your hydrophobic signal. And a part of the protein is exposed into the lumen, and there will be a short tail which will be in there sticking out. So you are looking at these other proteins. Now what happens, like you can see easily, that they form the vesicles, and these vesicles can form from the endoplasmic reticulum. Then they can go to the Golgi, and from the trans-Golgi network, they can also form these vesicles. These vesicles can walk by the help of the motor proteins on these, so this transport vesicle, and see that there is a microtubule, and they can be carried by the motor proteins onto the membrane. And now these vesicles also will have specific addresses, how to fuse with them. They are now fused with the plasma membrane. And see the interesting part. This is very interesting in the sense that the luminal side, the inside of the endoplasmic reticulum, that side becomes the exoplasmic side. So the proteins on the viral surface, they are facing inside of the ER, inside of the lumen, and they are exposed towards the outside. So basically the short tail that you have, all the proteins, which are sticking out in the cytoplasm, they remain as cytoplasmic tail. So we can say that in that sense, the ER lumen is equivalent to the exoplasmic phase or the extracellular space. So think about this way. So once they are going into the plasma membrane, you can see that there are viral RNA and other proteins synthesized in the cytoplasm. They can all be targeted and associate together with the membrane protein to form the mature viral particle. So these are basically the secretory pathway by which, generalized secretory pathway, we're going to see a specific example that how the proteins are targeted onto the plasma membrane. All right, so here what we are looking at, that the sub-assemblies are formed through an assembly line. And what happens that, say this for example, you can easily imagine that how complicated that process is. And there are formation of this discrete intermediate structure, and you cannot jump a step. They must be step by step. So when you're making the head or the tail fiber, it must go in a sequential assembly, sub-assembly. So it cannot proceed unless the previous structure is formed. So what happens, say for an example, you make the tail and the head together, and if you just do not add the tail fiber, or you just mutate the tail fiber, you will not, the virion particle will, mature virion particle will not assemble. But if you have this structure right here, which is the head and the tail together, and if you just add exogenously added tail fiber to that, it will assemble in there. But if you just take the head and then you just go and add your tail fiber, they are not going to add. So this must be formed before the tail fiber can be added, basically. So this is what it looks like. So now you can see that there are some arrows which are dotted, whereas there are some which are looking like they are straight dark, the straight solid arrows. The dotted lines are basically that those steps are irreversible. If something goes wrong, they can go back to the previous step and correct that and come back. Whereas the straight arrows, which are the solid arrows, they are usually unidirectional. So that's why they actually form this structure by the assembly line. And this ensures the quality control, that if the previous structure is not formed, the next structure will not form. So this is this is the reason that why these sub-assemblies are actually formed, and some of the steps are reversible just to have the quality control and the efficient process of making a correct structure. So here we are looking at some of the sub-strategies for making the sub-assemblies. So first, what you are looking at, here we are looking at from individual protein molecules, VP1, you have a pentamer, and in the middle you have either VP2 or a VP3. And see that this structure happens to be more towards the forward reaction. So see that this is a reversible structure, but see that reverse is a little bit dotted line. But this structure, when it's formed, does have a forward mode of forming these structures. So on the other hand, you can have a polyprotein precursor. This happens in poliovirus, which is a picornavirus, Picornaviridae family. So here, all you know that the viral genome is an mRNA. It comes into the cell and forms that huge long polyprotein. And you have seen that the different proteases process that. So once this polyprotein is formed, one of the portion of the polyprotein, VP1, 2, 3, and 4, VP4 and VP2 is together called as VP0. This is the P1 structure. Now what happens that with the help of some other chaperones, we are going to see that again in the next class, they are properly folded in this way. So VP1, 2, 3 like this, and the VP4 is usually going to be inside the viral particle in that. And when this VP1, 2, 3 forms, they actually make these, if you just remember, these beta barrel jelly rolls. Once that beta barrel jelly rolls are formed, at that time, the protease is a 3CD-Pro, which we recognize those specific structures. And you can see that VP1, 2, 3, they were initially covalently linked, but they are chopped off. Now they are not covalently linked anymore, except the VP4. So this is the polyprotein precursor, and this will be basically the 5S structural unit. So why we name them as a 5S? Because this is basically the sedimentation coefficient. If you just take the cell, lyse it, and centrifuge them down, this is basically that the 5 is in Svedberg unit, the sedimentation coefficient of this structure, which you call as a 5S structural unit, which will form again into a larger 14S subunit that we are going to see. This is, on the other hand, is a chaperone-assisted assembly. And here what we are looking at, that this is a protein 2, which is with the help of the L4, 100 kilodalton protein. This forms the adenovirus 2 hexon trimers. So you can see that the L4 is also an adenoviral protein, and that helps in the formation of the adenoviral hexon trimer. So we have seen that before here. You can easily see the L4 100 kilodalton protein. For the protein 2, it forms the hexon trimer, and then it is imported. So you need to form these sub-assemblies, and there are three strategies for that, for this process. The next part we are looking at, that as I said, that all these assemblies reaction are assisted by the cellular chaperones. So the first one that we are looking at as an example is an GRO-EL structure. So GRO-EL and GRO-ES, GRO-E large EL and GRO-ES is a smaller subunit. These are very well characterized chaperones in the E. coli. So it's a bacteriophage. So that's why it's a protein from the E. coli. Now, GRO-EL works with the association in the E. coli. GRO-EL works with the ES. Small and the large both together need to fold the bacterial proteins. Here, what happens that the GRO-EL associates with a T4GP31. This replaces the GRO-ES, and instead of helping the bacterial protein, it helps the GP23, which is a major capsid protein. It helps GP23 to form into the head, basically. On the other hand, we just saw that the VP1, 2, 3, 4 in the picornavirus, like poliovirus. Here it uses another protein in the mammalian cells, which is the HSC, is a heat shock cognate. It's a 70 kilodalton protein, and this is a chaperone protein as well. And with the help of it's ATP dependent, and you can see that this is basically folding in a proper way, and there will be also, along with that process, there is another additional viral protein P23, which I was talking about, folds these proteins into the beta barrel jelly roll. And once they are folded in that proper orientation, at that time the 3CD Pro can recognize the structures or the tertiary structures can recognize the structure and do the protease cleavage into the mature protein. So these are the chaperone protein, HSC70 and P23, which is a co-chaperone for the HSC70. Right here is the HIV or the retroviruses, and this is the tri-C, just like the GRO-EL is a large complex. So this is in the mammalian cell, this is in the bacteria. So basically what you have, that you have a top and the bottom structure, and there are octamers. There are 8 of these make these huge, large chaperone protein assembly. So what you are seeing, that remember that the genome of the retrovirus, they code for this GAG, then pol, then envelope region. So you take the GAG, once the GAG polyprotein, again remember that the GAG polyprotein is formed, it folds the polyprotein, the nascent GAG, with an ATP-dependent way, and these are the subunits from the GAG. So the subunits of these GAGs are the matrix protein. Here I wrote it down. It's a capsid protein, nucleocapsid protein, and P1 and P6 proteins. So, remember that there are 2 other proteins in there. Some of them are not in there. But those are the proteins right here. You can see that they are basically formed from the GAG. And how these are cleaved again. Remember that these are cleaved again by the proteases to form these individual proteins. And these individual proteins are assembled sequentially. You can easily look at that. There's a 10S subunit, 80S, 150S, 500S, and then finally the genome will be packaged inside the virion. So here also we are looking at the polyomavirus. We're constantly looking at this pentamer with the VP1 pentamer, and in the middle you have either VP2 or 3. This is also helped by the HSC70, the heat shock cognate, just like the poliovirus. But along with that, LT also help, and LT also help in the nuclear localization for as well, because the LT goes into the nucleus, and that is for the pentamers for the polyomavirus. So these are the chaperone-assisted assembly. So here we are looking at a particular way that how the poliovirus can be formed. We are looking at these proteins, 5S structural unit. We just saw that how it is formed. And from the 5S structural unit, you form the 14S pentamers. These pentamers, when interact with the genomic RNA, 14S pentamer with the genomic RNA, they will produce this provirion. This is still non-infectious. So inside the virion already assembled, these will lead to the VP0 cleavage of the VP2 and the VP4. So remember that this is your VP0, these together, the green and this one, is the VP2 and the VP4. And they are not cleaved yet in here. In here, when VP4 and VP2 are cleaved together, it will become a virion infectious particle, which basically buds out of that cell. There are certain lipid molecules which are associated with that, for these interactions. I've written it in detail, but again, the basic idea is needed. On the other hand, in an experimental basis, this could be your, some of your critical thinking questions. If you form these 14S capsid pentamers in an in vitro assembly reaction and you add the cell extract to form those, you will form those structures, 75S empty capsid. Now, when the 14S capsid pentamers are forming, At that time, you just exogenously add the genomic RNA, they will form the provirion. But once this 75S empty capsid is formed, remember that, once that empty capsid is formed, if you add the genomic RNA, it's not going to package that. So that's a dead-end product, basically. So you have to add the genomic RNA along with the 14S pentamers so that they can be packaged together. If the 14S pentamers do not receive the genomic RNA, they will form into the empty capsid and the process will be aborted, basically. So that's your sequential assembly of the poliovirus. Herpes virus are a little bit more complex, and again, remember that we'll not ask you individual names of these proteins, just the overall process is more important. So first, what happens that these are the VP5 pentamers, and if you carefully look at that, although this looks like the same, there are some dark ones. The dark ones are the VP5s, and the light ones are the VP6. VP5 hexamers, sorry. The dark ones are the VP5 pentamers, and the light ones are the VP5 hexamers. So initially what happens that the VP5 pentamers and the VP26, they basically form, remember that, VP5 pentamers, VP26, they form a protein scaffold. And onto that scaffold, the other proteins are gradually assembled. What are those? You have the additional VP5 hexamers, then you have the minor protein VP2319C. But initially what you are seeing that on this you have a pre-22A that is formed so that the whole structure can be supported. And along with that, inside the virion, this VP24 and 21 is also added, which are these rod-shaped structures that you are seeing. These are the proteases. And then finally, remember that the herpes virus has 12 vertices, icosahedral. In one of the 12 vertices, this UL6 portal is added. It's a dodecamer, 12 subunits. This is the portal through which the genome is packaged into the virus. It is only in one of the 12 vertices, and how it is basically determined is not very clearly known. So once this structure, you can easily see that you have the VP26 at the very top, then you have VP5 pentamer hexamers all in there. Now what happens right here, you can easily see that right here you have the pre-VP22A and other VP24, 25. These precursors basically mature, and the protease, VP24 protease, will digest all the proteins inside, making them empty. And basically the amino acids are going to escape through the VP6 portal. There is an opening in there, so the amino acids are going to pass away and make this an empty capsid. And the genomic DNA is going to be basically packaged inside, and we're going to see that how this happens again. So here we are looking at the concerted assembly. So here we are looking at sequential assembly, two examples. That concerted assembly, parallelly two pathways are going on and assembling together. So what we are looking at, that the genome RNA that comes in produces the mRNAs. These are going to produce these proteins like NEP, M1. Some of them get associated with the ER. They will form the membrane protein, the hemagglutinin, neuraminidase, and M2. Now, once the genome, these proteins are made, you can see that from the 3', 5' negative sense genome, they formed an intermediate plus, and these are the genomes that are made on the antisense plus genome. So these are the genomes, all minus sense. So these NEP and the M1, they are basically imported inside the nucleus. Out of that, M1 binds to specific sequences of the RNA. So it's an RNA binding protein. And on that M1, there are some sequences that is recognized by the NEP. That's the nuclear export protein. And once they are exported, this is a RAB, which is basically on the vesicle that brings it specifically to the membrane. So this is actually an address that where they find the membrane, basically. So this structure with the nucleocapsid enwrapped with the M1 is going to go to the membrane. And these structures, HA and M2, are also going to get to the membrane by the vesicle, and they are going to assemble and release from there. Remember that we are looking at this structure as a naked structure, but this Rab associates basically with an endosome and helped with a motor protein to walk in there. We are going to see the modified picture of this essentially a little bit later. So this is the pathway, this is a pathway simultaneously, concerted assembly, both go to the site of assembly at the same time on the plasma membrane. So we're looking at some of the feature of this HA protein, which is the membrane protein. So the HA protein is synthesized like with an HA0. This is the protein, membrane-bound protein, all together HA0. You have the signal sequence, which is chopped off by the signal peptidase. Always a quiz question that which cleaves the signal sequence, signal peptidase. This is not present in the mature protein. Now, there is also another thing right here. You have a fusion peptide, and this yellow dot is where a protease cleavage occurs. So that means now from the HA0, you have two polypeptide chains. One is HA1, this portion, this top portion. Another is HA2, which is membrane bound. Now, if you chop this off, how does this remain attached? Remember that you have a disulfide bridge right here. You have this disulfide bridge which keeps the HA1 and HA2 folded together and hide that fusion peptide. So here you are looking at the HA1 from the perspective. This is from the side view. This is from the top. This is the globular head. This forms the globular head, and this forms the stem of the structure. And the fusion peptide is buried in there, which will basically fall onto the membrane for the fusion. Here you are looking at the transmembrane domain that we were talking about. And this is that blue part that is in the membrane right here with the membrane retention signal that you are looking at. And then you have a short C-terminal end, and sometimes they are acetylated and they are buried back into the membrane as well. So buried back into the membrane. C-terminal really doesn't have much any more function other than that. So this is the viral protein that will bind to the receptor protein basically and help in the membrane fusion. All right, so here we are looking at that how this maturation of the HA0 happens. You can easily see that the protein is synthesized, and here we were talking about the C-terminal end has an acylation, and the acylation will bury that protein in the membrane back in there, so no open C-terminal. This is the HA0 with the intact protein, and as it is going through the stack of the Golgi, see in the trans-Golgi network, there is a cleavage that happens right here. You can see trans-Golgi network. So what happens? You have these two portions. You have an HA0 and, the HA1 and HA2 protein. They are separated out, but they are connected with the disulfide bridge. Remember that one of the confusion that happens, that when the protein is secreted inside the lumen of the plasma membrane, this is where the signal peptidase basically cuts the signal peptide off. This is not the signal peptide. This is your HA1 and HA2, and this is your membrane spanning or the transmembrane sequence. Here we are looking at the protease cleavage of the maturation of the HA0 to HA1 and HA2, basically, that happens with the protease cleavage. Now, next, what we are looking at, the genome packaging. Here is an example of the RNA genome, and they must be separated by the cellular DNA or the RNA molecule, and they must have this packaging genome of the in the viral genome as well. So let's take a look at that. Again, complicated pictures, but just a take-home message is easy. So here we are looking at the DNA genome. Remember that we are looking at not the protein, the DNA genome. In the DNA genome, look at this area that we talked about these, that you have an inverted terminal repeats. This is the 5' end of the adenovirus DNA. You have an E1A protein start site. Right here is a lots of enhancer sequence. But here also you see some AT-rich segments, which are basically recognized as a packaging signal. On the other hand, say for SV40, in the SP1 region also, there are some sequences on the DNA which are recognized by the cognate proteins for the packaging. And the take-home message that is complicated, just specific sequences are not required. The packaging signals near the left inverted repeat and the origin adenovirus. Signal is complex, a set of repeated sequence and overlapping enhancer that stimulate late transcription. So you can see that the same sequence right here in the genome, which earlier helped in the transcription, the same overlapping region sequences are also help for the packaging of the genome as well. And this is recognized by the viral protein 4A2. And we talked about that the 4A2 basically package the genome. And if you just remember that we have seen this in this picture, that how the 4A2 say, we are going to see that these hexane trimers are transported in there, and we are going to see that the 4A2 basically, which is also a transcription factor, will bring these structures together and they will assemble together. The whole structure is much more complicated, but we are just looking at that along with the 4A2, L4 is another protein which are also responsible for the packaging of these genomes as well. So that's it. I put it in the detail, but too much detail is not necessary. Just to understand the overall mechanism that the DNA genome packaging signals, where they are present. Many times they are just in the promoter region of the DNA. So here we are looking at the packaging signal of the DNA genome of a very complex herpes virus. So here we are looking at that this is a unique long sequence and the US sequence, and here we are looking at, this is an A sequence right here, followed by a B. Here is an A sequence, and before that you have a B and a C sequence, and here you have an A sequence with a C sequence right here. So what you are seeing that all throughout the genome, you have the first A, second A, and the third A. So the first A and the second A, you can have multiple of these sequence repeats in the first A and the second A. But in the last A after the CE at the 3' end of the genome, you just have only one copy of the A genome, or A sequence, only one copy. In the middle and on the 5' end, you can have multiple. And at the end, you have just only one copy. So what they are in there, so you have some direct repeats, direct repeat 1 and 2, and there are some packaging signals 1 and packaging signals 2. That is your A sequence. And you have you can have multiple of them in this position and in the middle, in between the UL and US, but only just one sequence in there. So what happens that at the portal where you have the UL6 portal, several different proteins assemble. And again, I'm just going to ask you individual names of the proteins, but just some specific ones we're going to see. These include EOL15, 32, and 28. And what happens that they recognize these sequences, these A sequences. When they encounter this sequence, they are going to package this genome inside, and you can see that this is basically spooled inside the genome. So see that what they're doing, that from here all the way up to here, you have one genome length of the genome that is basically packaged. Now what happens that these complexes will recognize several A sequences, several B sequences, and after that, once they get just one A sequence, they note that that is the end of the genome. They will cleave off at that point, and that will be just one length of the genome. Why this process is done this way, this complicated process? Because remember that the herpes virus genome is replicated by the rolling circle mechanism, and the genomes are synthesized as concatamers. One genome is connected to the next one, and the next one, and the next one. So this process basically is spooling the capsid full of one single length genome, and once they find the last single A sequence, they will just chop it off. And in this process, UL17 is a protein which is essential for the DNA cleavage, and another protein is also not shown in there, UL25. They actually withstand the packaging pressure. And this is an amazing part. Remember that the DNA is packaged into the herpes virus with 20 atmosphere pressure, 300 psi, 293 to be precise. So just imagine that 20 atmosphere pressure the DNA is put in there. So basically what happens, that UL6 portal, when it infects another cell, will sit onto the nuclear pore membrane, and it opens up the UL6 portal. The DNA is ejected out straight just from the pressure inside the nucleus for the delivery. So this is basically the packaging signal for the complex genome. Now we are looking at RNA virus genome, which is HIV. So here, this is what we were talking about that people get confused. Remember that. A portion is the RNA sequence. B portion are the proteins which recognize the specific portion of the RNA. So A is RNA, B is proteins. So let's start with the sequence. So here you can see that you have a TAR, the primer binding sequence, and then you have these DIS, DLS sequences. I have written all of them, that what they basically mean, but you just only have to find out just only one specific area. Right here you have this sequence which we call as a psi sequence. So the first thing that what is important in here, that this is basically a necessary sequence for the genome packaging, but again, not sufficient. You need other sequences, but this is where the packaging signal basically rise. That means that this is the portion of RNA which is going to interact with these proteins in here, or which is the GAG. So if this is your GAG, so you can see that this is your matrix protein, a capsid protein, nucleocapsid protein. From here you will see that P1, P6. So in the nucleocapsid protein right here, you can see that there are some positively charged amino acid, and there are two boxes with cysteines and histidines, usually 3 cysteines and 1 histidine. And this specific RNA binding site basically binds to the specific areas of the RNA to package them inside the virion as well. And here these are the different stem loops that we have seen. The SL1 is the kissing loop complex, which basically help in the dimerization of the 2 RNA molecules. See that they are connected right here, and this dimerization domain is done by these kissing loop structures, two of them coming together. So that's your packaging signal. Again, remember that I did not go into the individual of these one, but just the overview of that where the packaging signal lies, and to which is recognized by these nucleocapsid protein to enwrap those. Now, on the other hand, we are looking at this influenza virus. And in the influenza virus, think about that, you have 8 segments, and that's the problem. And if we just think about that, these genomes are basically packaged in a random manner, then you can have combinations of approximately 8 factorial over 8 to the power 8 different types of combinations that they can put in there. Because remember that you have 1, 2, 3, 4, 5, 6, 7, 8. And if you think about all the combinations that can be inside that, it could be that many. But that will basically produce viral particle, which is no good. They will not be infectious. It's a wastage of that product. So initially also we thought that how this process basically worked, and the idea of the packaging signal for these was not determined. It was just thought that they are packaged in random. But now we have the evidence of the specific packaging sequence of the RNA segments. These are the segments of the influenza virus packaging, and see that these are basically the proteins, and these are the number of amino acids which do contain the packaging signals. And we also know that basically the segments are arranged into this 1+7 configuration, and these are perpendicular from the budding site. So what we mean by this, basically, that this gray box that you are seeing, this is the side view of the plasma membrane. And what you are looking at, that as it is budding from the plasma membrane, all these individual RNAs remain perpendicular to that. So when you are looking at this picture right here, are the top pictures, we are looking at it from the top. If you look at that, so you have 1+7 arrangement. So this is fairly new, that this is only done by the RNA and RNA protein interaction, that how they are assembled together. This is your influenza virus. The selective packaging also, so this is basically the process of selective packaging, that you need only those 8 unique segments to be packaged inside the virus. More evidences of selective packaging comes from bacteriophage 5-6, and there you have 3 different length of RNAs. These are all double-stranded. They are small, medium, and large. And there is a serial dependence is required. You must actually put the small one in there so that the next to the medium and the large they are basically formed. So the scientists argued that if they are packaged these things randomly, then what will happen, your particle to PFU ratio will definitely not be 1. What we mean by the particle to PFU ratio is equal to 1 means every particle that you basically see is infectious. That means that each one of them must have a small, medium, and large segment. If you have 2 large, 1 small, or 1 medium, 1 large, like that, that particle will be non-infectious. So if selective packaging was not happening, then you will not see a particle to PFU ratio 10. It could be 10 or 50 or 100. That means you're producing 100 particles, and out of that, just only 1 is effective with the S, M, and L. Others are packaged randomly, the sequences. So these basically are the evidences that selective packaging basically occurs. So basic idea about the acquisition of envelopes. One of the example of that the glycoproteins and the nucleocapsid protein can basically help in the acquisition of the envelope. Internal matrix protein also can help in that retroviruses. We just saw the GAG polyprotein. And influenza virus, coronavirus, just the spike proteins or the hemagglutinin protein for the influenza virus, they can form the formation of the bud. And here in the, say for example, in vesicular stomatitis virus, the matrix protein can drive budding. So you can see right here, the matrix protein can drive budding. But if you have the glycoproteins, which are the red ones, and the ribonucleoprotein, it is much more efficient. So these are the several different types way that you can acquisition of the envelope, that how you can do that. So here we are looking at the membrane targeting sequence of the influenza M1 that we just saw, and also the VSVM. So and look at that this is a lipid binding RNP domain, and this is binding to the RNP, and you have these hydrophobic regions. On the VSV, you have these plus charges amino acid binding to the RNP, because remember that your nucleic acid is negatively charged. So that's why you have the plus signs in there for that. And right here you have the hydrophobic region. Hydrophobic region will interact with the membrane. So here we saw this picture once again. Remember that this protein right here, M1, this does have these hydrophobic regions and the lipid binding region binding to RNP. This is number 1. And also they have a sequence which can interact with the membrane for the packaging. So these all together, along with these proteins, can form the budding. So here is a little bit detail that we were talking about. This is the same thing, but just a detailed part that along with the M1 protein associated with the nuclear export protein, NEP, and then the XPO1 basically takes them out through the pore and associate this RAB onto the recycling endosome. These endosomes are brought into the cell, delivered, and they are now going back to towards the membrane for the fusion. And onto those, they basically bind with the RAB. And they will ride the microtubules with the help of the adenine motors to be delivered onto the membrane. And the rest of the part is also the same, which is not shown. So this basically is the same. But this transport is just not by naked part. They are always associated with the endosome and the microtubule transport. Here we are looking at the GAG retrovirus assembly. Here we already talked about the GAG part, and here you can see the matrix capsid, nucleocapsid P1, P6. These are the proteins. And then you have the GAG pol. So what happens that when you have the GAG polyprotein, this is produced, if 10 molecules, you produce 1 molecule of GAG pol. GAG pol will have all of them, but they will have the protease, reverse transcriptase, and integrase as well. So you will have the other proteins associated with that as well. But you need less amount of these proteins in the GAG, protease, RT, and these are enzymes needed in less amount. You need MA, CA, and the NC much more amount. They are produced in the GAG as well as the GAG pol. And these proteases will basically cleave these individual proteins in the form, and they will interact with the RNA to package inside that virus, basically. So GAG by itself can form the virus-like particle, and they are basically spatially and temporally correlated with that. So here we are looking at that how the matrix protein associate with the membrane. It contained a membrane binding assembly right here, which is basically an acylation. And we already talked about that this is the portion that binds with the RNA, basically. So if you remove this myristylation sequence where a myristic acid, which is a fatty acid added to here, they will inhibit prevent the interaction, and you will not be able to form the GAG proteins in there. So here we are looking at another part that this is the addition of the myristic acid. You can see that this is the myristate, and then you have any amino acids right here, and then some specific amino acid. This is added to the N-terminal of the matrix protein, and this basically allows this to be inserted onto the plasma membrane. So that's basically the overview of that. And the viral proteins are synthesized in the cytoplasm and modified by the lipids the post-translationally. Most of the times, the lipid insertions are like transmembrane domain. You have the hydrophobic sequence that are inserted in the membrane. This protein is produced that is the interesting part of the matrix protein. This protein is produced in the cytoplasm, and then just the addition of the fatty acid helps it to insert it onto the membrane. So very unique process. So we will, let's see what we have, how much we have left, 30. So we have a little bit of material left in here, and we'll stop here, and we will talk about just the release part, and that's it. Let's not rush. All right, so let's start. And before we start, just to let you know, I mean, like this is for many of us, I mean, like I don't see any improvement in the attendance as such, but this is for everybody. See, it's clearly what it means that some of the students who doesn't come to the class still can pull it off the exams. And it's up to individually, up to you whether you want to come to the class, what grade you want, or something like that. And I highly suggest that if your grade is not up to what you like to have, either talk to me and talk to your classmates that how you can do, form a study group and reread that how to be successful in this course. A 4,000, 6,000 level course will be significantly difficult. And again, the question that I just write all the details in the announcement that why you're concerned when your class average is 88% and 82%, as I say, it's like that's fine. But the thing is that if you are not achieving anything like that, I still need to take care of, it is not that I just only care for the top 40% or top 60% of the class. I just take care of or try to take care of even the bottom 20% or something like that. I hate to use that word, but that's what it is. And that's why I just try to just push you so that you do better. So as I said, that just stop by my office, let's have a plan, do your exam analysis. These are very important. You have these Excel sheets at the very top of your module, just below the syllabus. Take a printout and you will see that there are 4 columns that why you missed a question. Answer those questions honestly for all 50 questions that for your exam, and you will find a pattern. And that pattern will tell you that what you need to improve to study in the next pattern, so for the next test. And that is very effective. So do that, and you call that as an exam analysis or exam autopsy. That will tell you pretty much it. So that's about it. Let's start the material. So we stopped at the retrovirus budding, and here we talked about that this lipid moieties which are added to some of the proteins, and even in this case, we already covered this slide and the next one. And what we mean by this, that when you have this addition of the lipid to the viral protein, it allows the targeting to the membranes independent of the signal sequence. Now remember that signal sequence here, we are not talking about the N-terminal signal sequence, that is cleaved off. But the membrane proteins are already embedded in the membrane of the ER with an internal signal sequence that we talked about. I put that one in the video, the drawing that we did. And also I put a small, probably in about 9-10 minutes video at the top, that how these proteins are trafficked into the ER. If you have not taken cell biology or the secretory pathway, you have not gone through that. Just at least, if not the detail, if you can see that, that's great. If not, look at that 10-minute video that how the transportation of the type 1, 2, 3, 4 proteins actually happen, which are targeted into the cell. That will help. But even with that internal hydrophobic sequence, proteins which are embedded into the membrane, they are automatically trafficked to the plasma membrane. But here what we said, that basically the addition of the lipids to these proteins will help the protein to be transported and embedded into the membrane. So these are synthesized in the cytoplasm, modified in the lipids post-translationally. We usually do not see this in the host cell. This is very exclusive for the viral protein. So the next part that what we are looking at, that we call this as escort-dependent budding role, what is the end? So basically the escort is a huge system of more than 30 proteins. I have just written down the blurb in very detail just to have an overview of that. But this is basically called the escort, stands for the endosomal sorting complexes. Basically these proteins determine where things need to go. And here we have two examples. One is an HIV gag, and there's a protein P6 in there, and that we talked about that, but never talked about the role. It contains certain domains, and the other one is an MLV. This is a murine leukemia virus. Both are retroviruses. It contains a P12 in the gag. Remember that the gag is the matrix protein, capsid protein, and nucleocapsid. There are some spacers in here, and here this is also the gag. So the green sequences, there are 3 of the examples what you see. These 3 green sequences are called the late domain, or L. And these sequences have the signature for the budding function. L domains, and they are little bit different in that in the sequences. So you can see that there could be a proline, there could be a serine and threonine, alanine proline, all like this, and there is another one, PPXI. So they could come in different forms. So no matter what, just L domain, remember that, it could be in different ways. But what they do, that they will interact with the cellular protein TSG, and this TSG will be the adapter for the escort 1. And also the second L domain get associated with another protein called ALEX. The same way, on the other hand, this P12, PPXI, this motif also recruits TSG101 and ALEX. You can see that right here. And what happens, that the TSG101, ALEX, these complexes are then basically act as an adapter for the escort, adds to the escort 3, and basically they are all destined towards the membrane where they are needed to be budded. So here in this case, it is the plasma membranes. All these complex proteins along with this is transported onto the membrane. Along with that, there is also, in some cases, the gag can also have an ubiquitinated gag also are found. This ubiquitinated, or if you have the ubiquitination motif, those are also adapter for the escort 3. And what you are seeing in this picture, that you are seeing that a couple of viruses are, like quite a bit of viruses are budding, trying to bud out of the cell. But what happened that in the experiment, they have deleted these L domains. So you can see that these viruses can start budding out, but they are arrested in that state. The scission or the cutting off of the virus and egress of the virus will not happen. So that's basically the function of the L domain for these viruses. I have put it in very detail, but just the overview is basically needed for the whole function. The next part that what it shows right here is you can easily see that there are some of these matrix protein. These are ubiquitinated. Those U's are the ubiquitinations. And the escort-1 complex is like right here, TSG101, escort-1 complex is associated with that. And also the Alex is associated with that. And the Alex is basically at the ubiquitin. These are packaged into the virus. You can just find them in the matured virion as well. Now, along with that, the escort-3 is also basically finds its way basically in the neck region of the budding virus and the membrane. And this escort-3 complex is basically responsible for what we call as a scission or cutting off of the virus. When the cells basically divide, the escort-3 is also responsible for the scission of the two cells as well. And once this process is done, the escort-3 filament is dismantled and recycled back again. So basically what the escort pathway is that exosomes, there are vesicles which are released outside, multivesicular bodies where you have a vesicle inside a vesicle, or budding. All of these pathways are basically regulated by the escort pathway, and the viral budding basically employs this escort pathway by some specific domains, which is we call as a late domain or L domain. And that's basically responsible. VSP4 is another extra protein that is also responsible for the budding. Without that, the budding cannot occur. So again, remember that. The overview is good enough. Individual steps are not required. So here we are looking at the viral glycoprotein sorting in the intracellular membranes. Basically, the glycoproteins which go onto the membrane determine from where they are going to bud. What it means by that say for example, there is very highly studied in the Bunia virus, which is a hantavirus family. So you can see that there are two proteins in here. There is a GN, which is composed of G1 plus C, and then you have another protein called GC. So what happens, say for an example, both of these proteins, when present, they will bud out from the Golgi, or the formation of the budding of the virus will form in the Golgi. But remember that they are made into the ER and make their way onto the Golgi. If you do not have the GC, if you do not have the GC, you only have the GN, then the GN just basically accumulates in the Golgi. It will not be able to bud. You need both of these proteins to bud it from there. So on the other hand, say the coronavirus E1, they are that is responsible for budding of the coronavirus. They basically bud from the ERGIC, or which is called the endoplasmic reticulum Golgi intermediate complex. Herpes virus, we're going to see that they are the intermembrane space is one of the place from where they bud, and you have many other examples of these. And HSV GEGI helps them to bud out of the Golgi. So we are going to see that why we have HSV or the herpes simplex here and herpes simplex here. So we are going to see that as well. But basically, the take-home message is that the glycoproteins or the membrane glycoproteins determine which compartment the sorting is going to happen and the virus will bud out. It could be ER, it could be Golgi, it could be ER Golgi intermediate, it could be the nuclear membrane, and of course the plasma membrane that we were talking about. But other than the plasma membrane, there could be many other endomembrane places from where the viruses can bud out. So here we are looking at a very complex mechanism, and this is still a debate or controversy among different groups of scientists, but this is the overview of the latest accepted model. So the nucleocapsid is formed in the nucleus for the herpes virus. It's a DNA genome packed, and this is the capsid on top of that. It recruits two proteins, UL3431, and that helps the virus to bud into the internuclear membrane space. So just remember that. your nuclear membrane is a double membrane. You have a phospholipid bilayer, two of them here. You have two phospholipid bilayers here. You have membrane 1, membrane 2. This is the intermembrane space. And this is basically where you have a pore that is traversed by the nuclear pore. So the virus, along with these tegument proteins, you can see that there are lots of tegument proteins that those are associated on top of the capsid. They bud into the nucleus, and now you see that it contains the membrane right here. Interestingly, what happens, this virus again fuses with the outer membrane of the nucleus. You can see this is the inner membrane, outer membrane, loses the membrane along with some of these membrane proteins. You can see that these US3 or 51, these UL51, those are lost. But some of these associated capsid-associated proteins remain, and the virus comes into the cytoplasm back again, naked virus, as a naked virus. Remember that out of those associated protein, VP16, the transcription factor that we talked about, is among them. They are still associated with the capsid the same way. Now, it again goes into the trans-Golgi network, so that's why we had HSV right here, as well as right here. And here you see the GEGI, here you see that GEGI as well as MND. These glycoproteins are responsible for the fusion of this nucleocapsid along with these proteins into the trans-Golgi network. Once they are in here, it will bud out from the trans phase of the Golgi. Here you have the full virus with the membrane. You see that inside the membrane, and you have the membrane, the second membrane from the Golgi that will fuse with the plasma membrane, and the virus will egress with the membrane. Very complicated process, but you can see that it just goes from step to step, from nucleus to internuclear place to cytoplasm to Golgi, bud out as a vesicle inside the vesicle from the Golgi, which vesicle fuses and the virus is exocytosed into the extracellular space. Here is another example of that what happens in this trafficking process, and this is in the dengue virus, very interesting virus, and they do contain the type 2 glycoproteins on the top of them. Remember that the type 1s are the stalk, and then on top of that you have the spikes, trimeric spikes. They stick out like coronaviruses or influenza virus HA that we talked about. The flaviviruses actually do have the type 2, where these proteins or the proteins responsible for fusion lie on they have much more beta strands, and they lie flat on the surface of the virus. So how it happens, at pH 7.2 in the ER, these are the proteins which we called as the pre-envelope proteins. And there are two domains of them, and the red dots are the cleavage site for the proteases. So this virus buds out, and then it goes into, it fuses with the Golgi, cis Golgi. It goes to the trans Golgi, and in the trans Golgi, see that gradually the pH drops. That's the color, from 7.2 to 6.7 and to 6, pH 6. At pH 6, there is a conformational change on this envelope protein, and that exposes these protease cleavage site, and the trans-Golgi network of furin, which is a host-associated protease in the trans-Golgi network, cuts those domains out. The blue domains are cut. So this is a protease cleavage. But see that what is happening, that inside here, where you have the vesicle formed from the trans-Golgi network, the pH is 5.7, these two subunits are still attached. As the virus gets out in there, still attached, once it again goes into the neutral and slightly alkaline pH like 7.5, these two domains basically are separated from the envelope. So these two PR domains are separated out, and this is your mature type 2 virion. So you can see from the immature particle that after, even inside this, there is an immature particle, because these are bound onto the the envelope protein, and they are basically released, and now you have the mature virus particle. So this is a pH induced conformational change and maturation. Here we are looking at the vaccinia virus assembly. This is also another very complicated, and just make sure that you couple of key steps you very carefully note, and that is responsible. That will be helpful for your exam as well as for your quizzes. So basically, what happens that there is certain proteins. These are called the D15, D20 proteins. These are responsible for modifying these ER membranes, and they form this crescent-like structure. So this crescent is basically made from the ER membrane, and this enwraps the replicated DNA. Remember that for the pox virus, it brings its own DNA polymerase, and the replication of the DNA happens in the cytoplasm. So this whole thing is happening in the cytoplasm. They enwrap like this into an immature virion. And these scaffolding proteins that we talked about, these D15 scaffolding protein, they are responsible for making this virion in a brick shape like this. These scaffolding proteins are required, and these scaffolding proteins are not packaged into the virus. They are just in there for making this shape of the virion. This virion can be trafficked onto the plasma membrane and can egress the plasma membrane with just a single membrane. This is your number one observation. It can go out just with a single membrane. But there is another route, which is the most interesting part. These mature virion can be enwrapped with the post-Golgi or the early endosomes. Now, if you look at that, it does not go inside those, but rather enwraps themselves. So this will get elongated, enwraps themselves with the double membrane of this. So we carefully look at this picture. Inside you have a single membrane, which is basically this mature virion. Now here, if you are looking at, see that these arrows, this is two membranes, one with the blue dots, another with the red dots. So you have two membranes, and now when you have the wrapped virion, this basically contains three membranes, one for the virus and the two double membranes. This is important for your quiz or the exam, that in this pathway, the second pathway, the virus in the cytoplasm, if they are enwrapped with the post-Golgi or early endosome, they have three membranes surrounding them. Now what happens that they again can fuse with the plasma membrane, and they lose the outer membrane, and the two membrane remains, the original membrane of the virus, the mature virion, along with one membrane, but that blue protein, and you can see the red ones, which were the outer ones, that is basically lost. And this is the extracellular enveloped virion. So this is basically the overview of that it could be either wrapped with a single membrane or with a double membrane. And we are going to see that the actin tail basically helps these virus dissemination, and how this happens, we're going to see. It's a very unique process. All right, so here, what we are basically seeing on the left-hand side, you can see that these are the red part is the cell, and this elongated part are the extension of the cell, and the red dots are the viral particle, and we call this as a filopodia, F-I-L-O-podia, filopodia. And on the right-hand side, it's a structure that you can see that there is an actin tail, and on top of that, you have a vaccinia virus. The dot is a vaccinia virus. So basically, the viruses can egress through this filopodia made by actin tails, and basically it pushes itself onto the next cell. This is a process by which it is disseminated. So what you are looking at, so this is basically the structure on the right, that is your actin tail and the filopodia, that's what it is. Now, how the virus is transported onto the plasma membrane and how these things happen is very interesting. So here there are two dots. The red one is a protein called A36R, and the blue one is called a B5R. So this A36R get associated with the kinesin, and the kinesin will basically motor protein, and the kinesin will walk on the microtubule and it will bring it close to the plasma membrane. So here you can see that the kinesin, the A36R will get associated and the kinesin will walk. It will bring it close to the plasma membrane. And the virus will fuse with the plasma membrane. So basically, what is happening, you are looking at this structure that it will just bring the virus right here onto the plasma membrane. Now, at this point, what you can see that this B5R, this is exposed on the outside, and this will send a signal to various amount, a number of different protein receptor on the surface of the cell. These are host cell proteins, and that will basically activate the Src kinase. This is a kinase. This is a proto-oncogene as well, the Src kinase. The Src kinase will phosphorylate the A36R. Once it phosphorylates A36R, this is no longer attached to the kinesin. Instead, it will trigger the actin polymerization by this adapter protein like GRB2, NCK, and WASP, and ARP23 is responsible for the branching of the actin. So again, A36R, first role is the transport close to the plasma membrane. Second, once phosphorylated, basically recruiting the actin tail complexes on the plasma membrane, and this actin tails can push this virus. But basically, we are using the word, the propulsion of the vaccinia virus on the actin tails so that it reaches the next cells very easily. So this is a similar type of technique that is also used, although intracellular, by Listeria monocytogenes, if you have seen it somewhere else, riding on the actin tail or the philopodia. All right. So the last part that we are talking about is basically the release of the non-enveloped viruses. And we all throughout talked about the enveloped viruses, and these viruses are the naked viruses like adenoviruses or the polioviruses, how they do. They basically lyse the cell by apoptosis or the necroptosis. And there are many viral proteins which rupture the cell membrane, like there are viroporins, and these viroporins basically make holes in the cell membrane like the polyoma virus does, and the cell will lose its integrity and basically lyse and release the virus. And there are many different types of loss of membrane integrity with the inhibition of protein synthesis seen in adenoviruses. So this is a normal process, but there are some unique processes as well. This is most common. Basically, the cell lyse or burst and the viruses come out for the non-enveloped viruses. But there are some exceptions. In this process, non-lytic release of the non-enveloped virus, what happens, say for an example, for the polioviruses, the poliovirus basically make these double membrane vesicles, and on top of which the RNA is synthesized. And then what happens, that there are several proteins like these 2BC3A proteins, those induce the assembly of the virus on this double membrane and eventually enclose this virus particle in a double membrane structure. These double membrane structures are usually known in the cell as autophagy vesicles. Suppose your mitochondria or some other organelle is going to get degraded, they are enwrapped with the double membrane vesicle and then they are recycled. So the virus particles are inside those double membrane vesicle or DMV. We have seen that in the coronaviruses as well. We talked about that DMV, double membrane vesicles. The inner membrane is disintegrated, so you have just only one membrane left instead of a double, and this single membrane actually fuses with the plasma membrane, and the virions are released. So this is basically the overview of the double membrane vesicles formed by the autophagic vesicle. You see that in polioviruses as well. On the other hand, say hepatitis C viruses, this is a very unique process. The viral particles are associated into this multivesicular body. We already talked about this. This multivesicular body is a vesicle inside a vesicle. And you can see that each virus particle is enwrapped in a vesicle, and they are inside the inside a larger vesicle. The large vesicle basically fuse with the plasma membrane, and the viruses are released with these vesicles with the virus inside. Remember that. Here, there is a membrane outside the virus enclosing the virus, but it is not a viral envelope. The membrane is just a vesicle inside which you have a non-enveloped virus. It has nothing to do with the typical envelope where you have the viral proteins impregnated onto that membrane, which becomes an integral part of the virus. So there is no viral protein inserted onto that membrane. Now, what is the advantage of that? The advantage of that these vesicles can go in your body completely undetected, cannot be neutralized by the antibodies, because it is just a plasma membrane of your cell. All the proteins that are there are plasma membrane proteins. There is no viral protein on the membrane, and it can go undetected from the immune surveillance. And all it has to do that it goes to a susceptible cell, and that vesicle will fuse with the plasma membrane, delivering the virus inside the cell. So that's one of the typical way, or these are the two different ways that these are called the non-lytic release of the non-enveloped virus. It keeps the cells alive for a longer time, and they can release these viruses in this way. All right.